So in section 4, we're going to look at molecular evolution. And the starter question says to describe how duplicated genes form. And we're going to take a look at 3 different learning targets. We're going to look at molecular clocks and how they're used. We're going to look at how new genes evolve. And we're going to look at Hox genes and how they can be involved in evolutionary. So molecular clocks are a way we can compare the DNA sequences of different organisms and figure out their evolutionary relationships. So basically we can use the mutation rates to see how much time has passed between 2 different species or several species. The more mutations that occur, the more time has passed. This is taking a look at a DNA strand, and the highlighted or different colored areas are the mutations. We're going to talk more about this next. And each of these molecular clocks are going to tick at different rates or move at different rates. And this is because some of the DNA sequences will accumulate the mutations more quickly than others. But again, the more differences you see between the DNA sequences, the more time that has passed between the 2 species having a common ancestor. So if we look here at this diagram, you can see that B and C will differ from 2 different mutations, but B and C differ from A with 4 different mutations. So there's been more time since A has, or C, more time that has passed for A as opposed to B and C. B and C are more closely related. So there's less time since they share a common ancestor. Gene duplication. We know that during meiosis, chromosomes are going to pair up and swap genetic material. And this happens during crossing over recombination. We also know that sometimes when that happens, there's an unequal swap. So a chromosome can have an extra copy of 1 or even more genes. And a lot of times if those become mutated, it will not have an effect on the original gene. But over time, if that mutation occurs, it can evolve and it can modify the existing gene. The DNA sequences can change. This can happen when chromosomes break and reconnect, or when parts of it are lost, and also when parts are acquired from different viruses, bacteria, microorganisms, or other chromosomes as well. So a cell does not usually create new proteins from scratch, and we know this because genomes will use old genes for new functions. And we also know that when we look at gene duplications and rearrangements and how they contribute to evolution, they contribute to the variation of the alleles within a population. And this is what natural selection and genetic drift will depend on. So when we look at the development of genes in the body plans, it's known as evo-devo, or combination of evolution and embryonic development. These are looking at the relationships between evolution and development. And now we know that small changes in the master control genes are what are able to give us the evolutionary changes that we can see within the fossil record. So we're going to take a look at Hox genes and evolution. We know that small changes in the Hox genes during embryological development produce large changes in the adult animals. So when we look at insects and crustaceans, these are related in ancient common ancestors. And we know this because they have dozens of legs. Where today crustaceans, including shrimps and lobsters, have 5 pairs of legs, where insects have 3 pairs of legs. And we also know that each part of an embryo starts to grow at a certain time, grows for a specific time and then it will stop growing at a specific time as well. And these small changes in the start and stop times can make a big difference in the adult shape. So when we look at the long, slender fingers, that means there was a more of a period of growth. Where if there were short, stubby fingers or toes, there was a shorter period of